Hello, boys and girls. Welcome back to yet another physiology lesson with yours truly. This week, via Zoom, we are going to be talking about the components of blood and essentially the role that it plays in the renal and digestive system, how your blood plays a role in excretion, and a lot of other good stuff that goes along with it. So, this will be a relatively quick lesson. First, I'm going to talk about the contents of your blood. You might recognize this image as representative of a test tube. A whole vial of your blood placed in a test tube and then set in a centrifuge. See a similar pattern with plasma, your blood plasma, being isolated at the top, a layer called the buffy coat, which is the smallest of the three main layers, and then your erythrocytes, or your red blood cells, which are the heaviest, most dense, are going to settle at the bottom due to that centrifugal force. Red blood cells, pretty straightforward. These are the denucleated cells that carry hemoglobin, which is responsible for transporting oxygen throughout your body and dumping it onto your tissues at a capillary level as they become oxygenated and you're able to breathe and respirate and your muscles can contract. This buffy coat is essentially just white blood cells, leukocytes, very important for the immune system to function, your lymph action, which we'll probably talk about more in a future lesson. And then on top, your largest layer, but least dense, is your blood plasma. This essentially just consists of water, about 90% of it, as well as some proteins, mostly albumin and uh, immunoglobulin, I believe, and some fibrinogen and other trace amounts of proteins. What's important to know is that its blood plasma is essentially water, H2O. So that's an overall look at the composition of your blood, and we talked about the components of the plasma. So we're going to focus a little more on this part of the test tube, if you will, the erythrocytes. Erythros just means red in Greek. Gee whiz, fun fact. So the hemoglobin, which your red blood cells contain, undergoes this, I don't want to say convoluted process, but it undergoes a long and interesting process as it's converted into slightly different molecules through the use of enzymes in your body, uh, pretty much produced by the liver. You can think of that as your enzyme factory for all intents and purposes of this lesson. Your hemoglobin has four subunits, two alpha subunits. If you can just imagine the four-leaf clover, you'll have an alpha, you'll have beta, alpha, and then beta. And each of these subunits has a hemoglobin, or a heme, I should say, inside of it, which has an iron atom smack in the middle, which will have a very innately positive charge, Fe++, and it will bind to that free floating uh, diatomic oxygen in its gaseous form dissolved in your blood at the capillary level in your lungs as you're respirating, and it will transport the oxygen to all the tissues in your body that need it in order to keep you alive and avoid necrosis. So this HB represents our hemoglobin here. When it comes into, or I should say that your blood is created along with its hemoglobin, actually in your bone marrow. There's a process called hematopoiesis, which occurs there, and it's uh, essentially squeezed out of your bone marrow like a sponge. Very interesting process. So it's all starts in the bone marrow. So this heme oxygenase, which I represented lovingly with this acronym, converts this hemoglobin into something called biliverdin. Biliverdin, as the name would suggest, verd means green, as it takes on a bit of a greenish hue, and um, biliverdin in turn is converted into something called bilirubin via another enzyme called biliverdin reductase, which you can probably guess what it does by its name. Very straightforward. And the thing about bilirubin, as these enzymes are doing as enzymes do and committing these cleaving actions, sort of chopping up this initial hemoglobin molecule into different shapes. What you will see is, I think it was, don't quote me on this, these carboxylate groups that are sort of suspended on a hinge through these enzymes cleaving action. And in the form of bilirubin, they undergo this, I forgot what the biochemistry term was for it, but the principle that basically every polar molecule will hide its polar parts on the inside while its non-polar parts are on the outside in order to maintain a balance of entropy. I think it was called something like hydrophobic action or the hydrophobic principle. But in any case, these side chains, these functional groups on the bilirubin can swivel and they can hide these negative charges on the inside of the molecule and become nonpolar on the outside. And that isn't necessarily a good thing because when we have this sort of bubble with a lipid membrane on the outside, that means it can diffuse through tissues very easily. The principle is that large and charged molecules cannot diffuse across membranes very easily. And because bilirubin is relatively small and it's capable of hiding that charge, if given enough time, it will diffuse into your tissues. And uh, bilirubin, as you might have heard from a nat phys class, is what causes jaundice when it builds up in your tissues and that yellow tint gets reflected through your skin. That's not indicative of a healthy person. Otherwise, we would all be yellow. So to avoid the bilirubin from becoming diffused all throughout your tissues uh, from the liver, as represented here in the illustration, it becomes, it has these groups. I don't know how to describe them other than just calling them glucuronic acid. It sort of surrounds the bilirubin to create conjugated bilirubin, which prevents it from diffusing across. From the liver, the conjugated bilirubin will travel into the digestive tract through the jejunum and the ileum, the small intestine. And this glucuronic acid, the root word gluca, meaning sugar, will come into your intestine and the bacteria in there, such as E. coli, will digest that sugar and your bilirubin, which was before conjugated, is now converted into something called urobilinogen. Urobilinogen is nonpolar, but this time we're not so worried about its ability to diffuse across membranes because that actually contributes to uh, our ability to excrete waste. So as this nonpolar urobilinogen is able to diffuse across the membrane of the intestine and into the bloodstream, as represented by this artery here, it sort of tags along with all of the oxygenated erythrocytes, red blood cells, and it will be circulated back into the liver to become conjugated urobilinogen, and it will essentially just be recycled if there is an excess of them. this cycle will just continue and continue until some of it makes its way down the bloodstream flowing toward the kidneys. And this urobilinogen will become uh, simply urobilin, which is then later excreted out of the renal area through the ureter and the urethra as urine. But if this urobilinogen never diffuses across the membrane and simply keeps traveling down the digestive tract, it will be converted into something called stercobilin. And this is what takes on a brown color and is ultimately excreted as your stool. So you see, we started as red, turned to green, went to yellow right here with the bilirubin, continued down here, stayed yellow as urobilin was excreted, or eventually turned to brown with stercobilin. Very interesting process. So I'm going to move on to the last mechanism of our discussion for today. This is a pretty dense one. This is our grand finale. And I'm going to begin by talking about the three mechanisms that contribute to hemostasis, or the regulation of blood levels, your bloodstream, blood circulation. And the first of which, probably the most simple to explain to you real quick, would be vasoconstriction. Uh, for example, if your parasympathetic system is activated, your body's relaxed, your muscles don't demand nearly as much oxygen as they would in a fight or flight scenario. Uh, your veins, your arteries don't need to be open so wide. Your blood can relax a little bit. It can afford to move slowly. And that's a good thing, because if our blood was pumping super high all the time, our hearts would become super oxidized, and our lifespan probably wouldn't be near as long. We would live probably as long as, I don't know, a mouse, a kangaroo rat. The other two are mechanisms that are not so simple to explain. Hemostasis, regulating the levels of blood in our body. So I'm going to start with talking about platelet aggregation. Along with red blood cells, this is we're going to say this is like a capillary, perhaps, just running through our fingertips, delivering oxygen to the tissue cells on the outside. Here's a tissue cell. Here's the collagen around the capillary represented by this zigzag. And then here's our endothelial cells. As you might remember, we talked about last week, just a single layer of endothelial cells, very thin, very subject to damage, but in the trade-off, they are able to diffuse oxygen and nutrients very easily to feed our fingers so they don't turn purple and blue and black and fall off, which is what happens with frostbite. So our very thin layer of single endothelial cells releases VWF, or von Willebrand factor. I think I'm pronouncing that correctly. What? Willebrand? Von Willebrand? No, it's von Willebrand. Von Willebrand. Von Willebrand. So this factor, you can think of it as these tiny particles that are able to bind platelets through this receptor that's called PB1 or GP1B. And these, all that's important to know about them is that they're mechanoreceptors. Once once von Willebrand bonds the mechanoreceptor site on the platelet, nothing happens. It's essentially inert. But let's say your endothelial cell membrane here is damaged for whatever reason, like you pricked your finger to get uh, to test your blood before you donate for the Red Cross or something. Well, your platelet will naturally sort of float towards this trauma site by which you just pricked your finger, and the von Willebrand factor that's connected to this platelet will bind very strongly to the negative charges of the collagen that are exposed, that are no longer insulated by this endothelial cell membrane. I call it a cell membrane when it's really just the walls of your capillary. So as the von Willebrand binds the negative collagen and the platelet is attempting to still float along in the bloodstream, you get a little bit of a twisting motion, and the mechanoreceptor activates because it senses movement. It senses a kink at the connection point, at the receptor connecting the ligand and the greater platelet molecule. And this, my friends, is what activates the platelet and causes it to take on its bizarro monster form. This innocent little oval becomes an eldritch horror, and it develops all sorts of interesting quirks. It has these alpha granules, I believe. Yeah, alpha granules and dense granules. Alpha granules and dense granules, they were latent, they were present in the platelet in its original form. They are only released in its activated form on binding this platelet or this platelet VWF collagen complex, if you want to call it that. But the point is, these alpha granules release more von Willebrand, so naturally you'll see a big buildup of von Willebrand ganging up on this collagen site, which will bind more platelets. As far as I'm concerned, the platelets and the von Willebrand, it doesn't matter who binds who. It seems like, for all intents and purposes, it doesn't really matter. These dense granules release ADP, which activate uh, platelets via a receptor site called something very long-winded. Oh no, not this one. It's PYR12. The long-winded one is what binds the the receptor site that I'm going to talk about next. PYR12 receptors are also present on the surface of platelets like this one. They bind ADP and they become activated in that way as well. In the membrane of the platelets, there is TXA2, stands for thromboxane uh, prostaglandin. There's yet another receptor for thromboxane prostaglandin when bound on the surface of the platelet, activates it again. And one thing that is unique to the activated platelet is this very special receptor, which is called like GP2B3A, or simply put, the fibrinogen receptor. These are only expressed and present on the active form of platelets. And their job is to take latent, floating around, free floating fibrinogen in the bloodstream and to bind it and to bind to another platelet, connecting them kind of like Legos until a giant mass of platelets is formed, plugging up this trauma site around the collagen. So there's our platelet aggregation. I'm going to go into a little more depth about the factors that go into this whole process of clotting up. And this is called the coagulation cascade. So we have two separate cascades that sort of converge into one called the common pathway. But they start off, they they can start one of two ways. Through the intrinsic pathway and the extrinsic pathway. We're going to start with the intrinsic, the more complex one first. So each of these numbers that you see might seem a bit convoluted at first glance, but it's quite simple. We're going to call each of these numbers factors. This factor 12 goes by the name of Hageman factor, and as it's exposed to the negative charges, it reacts similar to the way that von Willebrand's does. This Hageman's factor becomes activated into factor 12A, A meaning activated. And this activated factor is an enzyme that cleaves yet another factor to become its own active enzyme. So 12A cleaves 11 to become 11A. 11A cleaves 9 to become 9A, as well as 8, factor 8, which cleaves factor 10 to become factor 10A, and factor 5, which cleaves factor 2, which is called prothrombin. Yes, prothrombin becomes cleaved to become 2A, its active form, which is thrombin, which then finally cleaves factor 1, which is called fibrinogen, and is cleaved into its active form, that is fibrin, as well as factor 13, which doesn't have a name, at least not one that we're worried about right now. Fibrin, you would recognize under an electron microscope as being like this wiry mesh that forms a plug over this collagen. So if you've ever picked off a scab and noticed that it had sort of a fibrous texture to it, congratulations, you discovered fibrin. Nobel Prize. Another way, so we just covered the intrinsic pathway, called intrinsic because it's the collagen itself. It's this negatively charged quality that's intrinsic that is responsible for this scenic route that the cascade might take. What about the extrinsic pathway? Extrinsic refers to the tissue cells that are on the outside of the collagen, and all of these threes here represent factor 3 or tissue factor, which leaves factor 7 into its active form of 7A. Doesn't have a name, and 7A will converge once again onto the common pathway for the coagulation cascade. So beginning at 10A being cleaved into, or yeah, 10A and 5 being cleaved from factor 10, that's where the common pathway starts. It doesn't matter how it begins, it will always end with prothrombin, thrombin, fibrinogen, and fibrin being created in that order to ultimately clog up the mesh that you made. And that's important to know because if we are ingesting chemicals that damage our endothelial walls, or if we have a plaque building up within the collagen layer due to ascites, eating a bunch of junk food, fried oil kind of junk, uh, the more likely that these endothelial cells are going to be pushed in the way of the bloodstream, and from the sheer hydrostatic pressure, endothelial cells are going to get sheared off, and you're going to see clots being built up in the capillaries and in the arteries and the veins where they're not needed at all. You're sustaining unnecessary damage due to a problem that's totally preventable. And heaven help you if that giant clot that forms in the middle of your artery comes loose and goes up into your heart and lungs, and you have a myocardial infarction, pulmonary embolism, heart attack. Any way you slice it, it's not good, and people have died from it before. Well, we're pretty much at the end of our video here. I'm going to go over some very quick uh, three more molecules. So TPA, or tissue plasminogen activator, kind of similar to our von Willebrand factor here, is released in these little particles, these little granules from our endothelial cells. And this tissue plasminogen activator, I don't know how to explain it more succinctly than its own name does, but it activates a molecule called plasminogen that also comes from the liver and is cleaved by yet another enzyme to become plasmin. Plasminogen generates plasmin, and the essential purpose of plasmin is to degrade fibrin that forms these clots. And so the main purpose of plasmin in this case would be once your endothelial cell walls have been healed up and the clot is no longer needed, plasmin will be released in increasing amounts until all that fibrin is degraded and returned to the bloodstream in its inert state. So our intrinsic pathway, just to close things off, is um, to prevent clotting or coagulating from beginning in the first place. Heparin is a medicine that would be applied to prevent 12 from being cleaved at all, or even interacting with the collagen. Heparin, or Coumadin is its other name. As for the extrinsic pathway, that would be blocked by, well, excuse me, it's not, heparin is not another name for Coumadin. Coumadin is what acts on the extrinsic pathway, prevents factor 7 from being cleaved by tissue factor. It's Coumadin or warfarin. So that's uh, two separate medicines that perform a similar role, not identical, but similar to plasmin. It's these anti-clotting factors, you could think of them as. Well, everyone, I hope you enjoy this video. I tried to keep it as short and sweet as possible, as I always do, but my prayer is that you come away from this understanding a little more about physiology of your body and that you appreciate the grand design. That's all from me for this week, and I look forward to talking to you all again. Thanks for listening.